Okay, so successfully we have completed meiosis 1 and you people have understood meiosis 1 properly. I hope so. So what we saw in meiosis 1, we will see that in meiosis 1, what had happened? The cell had nucleus, had nucleolus, and it had 4 chromosomes. For our convenience, we have taken 4 chromosomes. So these were the 4 chromosomes. Okay, these were the 4 chromosomes. They were somewhere else, wherever they want, right? They were like this somewhere. And then one more we need to draw. Okay, this is how you found that there were 4 chromosomes in the start, at the start. Okay, at the start of what you can say, meiosis 1. I mean, in prophase, metaphase, so many things happened. Everything we have discussed. I'm just telling you what happened from how. So, meiosis 1, this was the condition. Later on what happened was, at the last, we got 2 cells out of this. And what you can say, the 2 cells which we got from meiosis 1, they had the nuclear envelope also. They had nucleolus. They had nucleolus. Complete only you can show, no problem. Okay, but the chromosomes, what they had chromosomes like this. They had chromosomes like this. Okay, and here, yeah, one is here, you know. Okay, and they had chromosomes like this. I mean, for your understanding, I'm telling you, I'm just telling you. But all the chromosomes what we have here, they all are different. Why? Because they all have exchanged the segments. So they all are different. So this is the product of meiosis 1. From one parent cell in meiosis 1, we formed 2 cells. And here the chromosome number is 4. And here what you can see, the chromosome number is 2 and 2. So we have already completed meiosis 1 in the previous video. Please watch it. You will understand. This is for your convenience. I am just trying to show you. From one cell we have formed 2. But all the 2 chromosomes what you are seeing in these 2 different cells, they are different. So you cannot say that they are similar to the parent cell. These have got different chromosomes. These have got different chromosomes. Among that also, they are not purely paternal or they are not purely maternal also. Completely different chromosomes, right? Because of the exchange of segments, where this segment exchange and all these things happened in prophase 1 of meiosis 1. And that also we have different stages. We have understood all that. So what we will see now is we are going to understand meiosis 2. Fine. So the blueprint what we have, that also we need to understand now. Fine. So what you can see here is that according to the blueprint, we have completed this. That it had 4 cells, it completed forming 2 and 2. This is done. Now what you will see, now we are going to see from these cells, we have already got 2 cells in meiosis 1. Now we are going to divide these 2 cells and we are going to form how many? 4 cells. What will be the chromosome number? Same. This is what we are going to see now. So this is meiosis 2. Fine. So we will just remove this because we have completed. So we are going to start from here. Just this much we are going to see. This is the blueprint for now. So let us start meiosis 2. We are going to discuss about meiosis 2. One more thing I would like to tell you. I am not going to go in detail with meiosis 2. Why? Because meiosis 2 is exactly same like mitosis. So no need of much explanation. Just like a crude thing we can understand. So we have already got in meiosis, again we know that there is karyokinesis and again cytokinesis. But karyokinesis 2 and cytokinesis 2. Yes or no? It has 2 things. First thing is karyokinesis 2 and then cytokinesis 2. And what about karyokinesis 2? In karyokinesis 2, we again have what? Again have prophase, metaphase, anaphase, telophase, right? So we are going to see that we have prophase 2, metaphase 2, anaphase 2, and finally we have telophase 2, and then we have cytokinesis 2. Okay, so see here, these two cells I'm going to draw here. In prophase, what will happen? We have because we have come to meiosis 2 because we have completed meiosis 1. So we are going to start meiosis 2 with how many cells? 2 cells, right? So what will happen in prophase? Again, the 4 important key points what I have told, that will remain the same here also. We have chromosomes like this. We have chromosomes here. We have chromosomes like this. Okay, but they have the segments of what? Like this. Okay, and what about this? We have chromosomes like this, and one is like this, but we have segments of this. Okay. Now, in prophase 1, you know that there will be nucleus. In prophase 1, you know that there will be nuclear envelope and there is nucleolus. Here also there is nucleus, there is nuclear envelope. What will happen in prophase 1? Nuclear envelope will start disintegrating slowly. Yes or no? This is what is happening here. Nucleolus also will start disintegrating. Here also. Nuclear envelope is disintegrating. The points are same, so I am not writing. If you are not understanding, please watch the mitosis video wherein you will understand these key points easily. So here, what is the points of prophase 2? For both the cells, what is happening is nuclear envelope disintegrates, nucleolus disintegrates, and condensation of chromosome. Again, because once the meiosis 1 has completed, cytokinesis 1 is completed, the cells will remain silent for some time, and that phase is called interkinesis. It is not interphase. Please, I have already discussed this earlier. Don't get confused. After meiosis 1, before entering meiosis 2, the cells will take little bit rest. That is interkinesis, not interphase. And what happens in interkinesis? Nothing will happen in interkinesis. It is just a stage of waiting, since it will start for the next one. Okay, nothing will happen much there. Little bit any repair or anything, all those things will happen. That's it. No replication of DNA. No, nothing will happen there. Okay, after that suddenly it will start with meiosis 2, and that too karyokinesis 2 in that prophase 2. What will happen? The thin, because after cytokinesis we had seen the chromosome were in the form of thread. So that condensation of chromosome will start in what? Prophase. So in prophase, nuclear envelope disintegration, nucleolus disintegration, spindle fibers formation also will start here only, because the centromeres again they will divide. They already had centromere equal. They divide and they start forming spindle fiber like this in the prophase. Right? What will happen in the metaphase? You already know that in metaphase the chromosomes will be brought at the center. This is what happens in the metaphase. Even if it is, what is the difference between metaphase 2 and metaphase 1 of meiosis? Their bivalents were there at the center. They had not moved away. Here, in this metaphase, you will see that these chromosomes have come at the center, equator, where it is. Yeah, here only. Okay, they have come to the center of this one. Now what will happen? also you will see that in metaphase, spindle fiber will get attached to the kinetochore. This is at least you know, right? So here also, spindle fiber will get attached to where? To the kinetochores. So this is metaphase 2. But what is the difference is that in metaphase 1, bivalents were there at the center, but here in two different cells, you can see the chromosomes are at the center, but the chromosomes are different chromosomes. Means they are variety now because they have got the segments, exchanged segments. What will happen in anaphase? Okay, so in anaphase, again two cells we are going to show. I am simultaneously showing what is happening in both the cells because we are talking about these two cells, no? So this and this. Now, so what you can see in anaphase, the centromere will divide. Here, key point of anaphase 2 is centromere will divide. In anaphase 1, no centromere division. Please note down these things. So here what will happen? The centromere will divide. So what is separated now? In this you are going to separate the chromatids like this. Try to understand. You are going to separate the chromatids like this. And they are moving towards the end. Like this. Separation of chromatids is happening in this anaphase 2. Right? What about this? Again here also you can see the centromere will divide. That is why the chromatids will move towards the pole. Because of centromere division. Like this. But what you need to show again? You need to show the segments. The segments are here. In this and this. This and this. What about this? They are somewhere here. In this. So, the varied chromatids you can see here. Right? So, this is anaphase 2. Now, what will happen in telophase 2? You know that in anaphase, they will move towards the pole. Yes or no? This is also okay, like this. They are moving towards the pole. So then what happens in telophase? Okay, now there is no space for me to show this. Okay, can you please, you have written in your books, no? I will please rub this because I don't have space. So, in telophase, you know that from this cell, what is the main feature of telophase? You people know very well. So in telophase, what will happen? These chromatids will move completely to the pole in the anaphase itself. Yes or no? Then, okay, that also I'll show here only. So this is early anaphase. What will happen in the late anaphase? Late anaphase, you will see that. Late anaphase, you will see at the end. Chromosomes at the end. What about here? Here also you will see. Not chromosomes, sorry. Chromatids at the end. And some chromatids have this, what is that? Segmented, this one, this side, like this. And what about this? Late anaphase, if you want to show, then you will show them towards the end. So this is the thing. And here also we have the chromatids, separated chromatids. And where is the segment? Extra segment, what they have got is here somewhere. Okay, so I hope you understood. Varied chromatids are there. What happens in telophase? In telophase, they start, I told you, telophase is again, it is always what? It is called as opposite of prophase. So what will happen? In prophase, condensation had happened. In telophase, this chromatid becomes loose like this. This also becomes loose like this. But what is you should show is this part has that exchanged segment. Okay. Now what about this again? In this cell also this becomes loose like this. Condensation, and this also becomes decondensed. But what you need to show? You need to show that the segment exchange is somewhere here, somewhere in that. So this is telophase 2. And then what will happen? Nuclear envelope will start beginning again. Yes, nuclear envelope. Because telophase 2 is opposite of prophase. So in prophase, nuclear disintegration had happened. Now here, reappearance. Then again, nucleolus will reappear. All the 4 nucleolus. And already here there was spindle fiber, and you will not see spindle fiber in telophase 2. Why? Because in prophase 1, spindle fiber had, prophase 2, spindle fiber formation was done. So in telophase 2 ulta, spindle fiber will be removed out. Finally, so this is telophase 2. Fine, please take down this much, and then we will discuss cytokinesis. Fine. I hope you understood karyokinesis too without any problem. So let me rub this. So we have completed successfully karyokinesis 2 of meiosis 2. And then we are going to see now cytokinesis. So I can show it here only with this diagram. In cytokinesis, you know what happens in cytokinesis. Okay. I'll just shift this diagram this side, okay? I'm just going to do that. So what will happen? Here I'm going to show that. Those were the 2 cells. I'm going to show it here. So in one cell, this was one cell, and nucleus was formed, both the sides, and what we saw is there was decondensed chromatid, we need to use a different color here. Yes, which one had that change? I forgot that. We will show it somewhere here. I forgot. Please see, somewhere it will be here, and here it is somewhere. I hope so. Here. Okay. Now what will happen? So this is telophase 2. Now in cytokinesis, you know it is furrow formation. So the furrow will start forming. Because this is, I am showing animal cell. If it is plant cell, what will happen? Again I am repeating, it is cell plate formation. So in animal, this is animal cell, so I am showing you furrow formation. So this happens like this. Here also it happens like this. Finally what will happen? Slowly, gradually the furrow will deepen. Finally it will touch. And then what will happen is, completely it will form a new cells. Like this. Are you understanding this? So, sorry, I did not show you the nucleolus here. Okay. Now, what you can see now, this becomes one cell, this becomes one cell, this one, this and one. So, I'll show you how. See, all the 4 cells we will see. Cytokinesis 2 leads to formation of 1, 2, 3 and 4. And you can see that this is one cell, this is one cell, and this is one cell, this is one cell. What about the nucleus? Yes, all the cells have got their nucleus, their nucleolus, nucleus, nucleolus, nucleus, nucleolus, like this. What is the difference? Here it has a variety of genes. This has some variety of genes, like this. Just to show you the variation. So I hope you understood. We have got 4 cells at the end of cytokinesis 2. So we have proved this, what? Blueprint. What was the blueprint? We need to show 4 cells with, this is the blueprint what in meiosis 2. So we started with 2 cells, we ended with how many cells? 4 cells. And all these 4 cells we call them as gametes, and this is the end of meiosis 2. In that also cytokinesis 2. Once the cytokinesis 2 is completed, even if it is plant cell or in plant cell, plate formation, in animal cell, furrow formation, finally leads to how many cells? 4 cells. So in meiosis 1, we proved this one. We proved this one. In meiosis 1, we proved this. And in meiosis 2, what we did? In meiosis 2, we proved this one. Yes or no? If you are not understanding, please watch the video from part 1. I have mentioned it as meiosis part A, part B, part C and part D. Okay. So please watch all the parts. Then only you will understand meiosis properly. Please take down this much. And then we came to the end of the chapter. We need to discuss about what? We need to discuss about the significance. What is the importance of meiosis? Successfully, we have completed meiosis 1 and 2. I hope you people understood this properly. Still, if you have any doubts, again and again you watch the video, it will be cleared. Still, if it is remaining, then please contact me so that, or comment, I can answer the doubts. Fine. So we are going to talk about meiosis significance so that we can end up with a chapter. What is the importance? What is the significance? Why this meiosis plays important role? Significance of meiosis. See, try to understand. First of all, the first point in significance is you only saw meiosis 1 and meiosis 2. And in meiosis, you saw what all happened. Okay. First thing is important in this meiosis is maintenance of the chromosome number. Means maintenance of chromosome number. See, if this meiosis would not happen, what would happen? If this meiosis would not happen, then the gametes would have the same number of chromosomes like parent. Then that gamete, when it fuses with other gamete, it will form double number of zygote. A double number of chromosome will be there. Triple. It will start increasing. No, are you trying to understand? Again, already I have discussed this, but still I am doing it. The parent cell, for example, let us consider this is the parent cell. It will first form the gametes. No. Human cell, if you consider, this is spermatocyte which has 46. It needs to form the sperm which is 23. Because of meiosis, it will form 23. But if meiosis would not take place, then it would be 46 only. Let us consider in females. In females, it would be 46 of oogonia cells. Then it will form ovum. It should form ovum which will have 23. But if meiosis would not there, then it would also have 46. Then what would happen after fertilization? Zygote would have 92 chromosomes. Are you understanding this? Now this zygote, if it is a male or a female, it depends. This zygote will develop, it becomes baby, it becomes adult, and when it starts producing, if it is a baby boy, it will start producing sperm. Even there, if meiosis is not there, the sperm will have what? 92. And if it is a baby girl, it would produce 92 in egg also. And those egg and sperm, if it gets, I'm talking about like fertilization, usually the process of fertilization. If it happened 92 plus 92. So likewise, it would have increased, like that. So if that happens, what will happen is, if the chromosome number of the species is not maintained, then it will not remain as that species. So many things can happen if that happens. So meiosis is very much important to maintain the chromosome number in all the organism. This is one thing. Second thing is, meiosis, why it is important? Because formation of gametes. Formation of gametes. And if gametes are important, no? Why? For sexual reproduction. So formation of gametes plays very important role. Why? For the sexual reproduction process. So that gamete formation is happening because of what? Because of meiosis. Then the third thing is you saw that in meiosis 1, the leptotin, zygotin, pachytin, diplotin, dikinesis, because of those stages, we got a variety of chromosome. So that leads to what? That leads to variation. Yes or no? That is why all the 4 sperms what we get, or all the eggs what we get from this process of meiosis, it will not have the same type of chromosome of the parent cell. That is why we all are different. Even if we are siblings also, we are different. Why? Because we are not exact ditto like our parents, because we have got different gametes. And these things are complicated. In case of twins, people will have doubt. So that we are going to discuss when that concept comes. So there it is a different thing. But normally if you see, what will happen is, we are not exact copy of our parents because of sexual reproduction, because of gamete formation, and because of this meiosis 1, particularly prophase 1. Clear? So this variation what is there? This is very much important. Why? Because of this leads to what? This leads to evolution. So variation is important, and variation happens because of meiosis. What else you require? This is enough. What all we are seeing, variety of species of plants we have, variety of species of animals, variety of species in case of every living organism is because of meiosis. Because of meiosis. If meiosis would not be there, or if it is only mitosis, then like organisms which undergo asexual reproduction, they doesn't undergo meiosis, right? They just go through mitosis. That is why they all are similar. They all look similar. No genetical variation at all. Because of meiosis only, because of sexual reproduction only, we have variation. Clear? So this chapter is completed. Okay, and we need to solve the questions. We will be doing that. I hope I have made all these things clear. Please, if you have any doubts, please ask me in the comments so that I can explain again. Okay, take down this much. Thank you.